Friday. Friday good. Saturday better. Unless you got a busy Saturday. I've got a very busy Saturday, so maybe Friday's better. I don't know. So, a couple of things. One, I apologize. I haven't gotten the old exam posted. I will do that this evening. If not, you may do something really nasty to me. Okay? I will get it posted. Number two, we have an exam in here a week from today. That's good news, right? No? It's good when you're done because then you're done with the exam and it's Friday. So it's like a win-win, right? Okay. I've had a couple of questions from students with questions about, well, how do I calculate protein charge? Because I haven't gone through that this year. I usually go through that. Okay? And what I've done this year is I've made a video where I describe how to calculate protein charge. I'm going to post that video tonight along with your exam, and it will tell you how to calculate protein charge. You are responsible for that. Okay? I'm going to tell you very briefly as I start, because protein charge came into play when we talked about the different gels and separations that we were doing up here. And calculating protein charge is something that you should know how to do. Okay? Now, I find students sometimes find that those descriptions are difficult or whatever. Please feel free to come and see me. I'm more than happy to meet with you and show you if my video doesn't help you. Okay? I'll tell you a couple key points to think about when you're calculating protein charge, though. Then you can watch my video, right? Number one, remember that when we have a polypeptide, the only alpha carboxyls and alpha aminos that are there are the ones on the end. The one in the middles disappear, and they don't contribute to the charge. That's number one. Number two, yes? I'm sorry? Would I repeat that? Yeah. So when we're looking at a polypeptide, we have all those different individual peptide bonds. Those individual peptide bonds are gobbling up all of the internal alpha aminos and alpha carboxyls. So the internal ones don't affect the charge because there's no charge associated with them. Only the ones on the end, the very first amino and the very last carboxyl. So that means when we go to calculate the charge of a protein, we have to find the charges of the following things. The alpha amino, that is the very first one. The terminal carboxyl, the alpha carboxyl. The ones in the middle don't matter because there's none of them. And the charges of any of the R groups that ionize. Any of the R groups that ionize. That's the three things that will contribute to the charge of a polypeptide. We need to know those. Okay? Now the video I will post will tell you how we do that based on what the pH is and what the pKa is. Okay? Yes? So the question is, do amino acids in an alpha helix always have a neutral charge? The answer is no. The charge will always depend upon the pH and the pKa of the group. I'm telling you, that the only way that you can make that determination is if you know the pH and the pKa. So it depends on the group. There's no one set answer. You have to calculate it each time. Is there a formula that will tell you, and that's what I'm telling you? Yes. The pKa and the pH will tell you. All right? I'll give you a quick rule if you'd like a quick rule. This one I won't give you on the exam, so you'll need to know this if you want to know this. Oh, where was going to write now? Okay? Here's how it goes. If the pH is more than 1 unit, or let's say 1 or more units, below the pKa of a group, if the pH is 1 or more units below the pKa of a group, the proton is on that group. If the pH is 1 or more units below the pKa of that group, the proton is on that group. On the other hand, if the pH is 1 or more units above the pKa, the proton is off of that group. Okay? So that's the formula I think you were after. Is that it? Okay. Now, you will apply that rule, and that video I'm going to post will show you on a polypeptide how one would go about determining the charge of the groups at a given pH and all the pKa values. If you want to memorize the pKa values, I will give you those on an exam. I'll give you the pH, but you will have to calculate the charge of one. Okay? So if you watch the video and you're still confused, then come see me, okay? Yes? So it depends on whether it's negative or positive as to what the group is. So a proton on an amino is a positive. A proton off of amino, as we talked about the first day, is 0. A proton on a carboxyl is 0, and a proton off is minus. So it depends on the group. So you need to go back and review what I talked about the first day on that, okay? Okay. So please look at that, and I think that will give you some assistance in understanding that process. Okay. So as I said, I'll post exams and I'll post that video tonight. Please look at those and you'll be set. What I want to do today is briefly finish up talking about techniques for isolating proteins. And then what I want to do is move on to talk about the cool subject of enzymes. Okay? So, last time we got into some cool stuff. We talked about how it is that we can separate proteins. All right? We separate proteins by coating them up with this detergent called SDS, making them look like they're a large, negatively charged rod, and then we use an electrical field to push them through an acrylamide gel, and they race through, the fastest ones being the smallest ones, and the slowest ones being the biggest ones. Okay? All right. Now, I'm going to tell you about another cool technique that combines that with yet another technique. It's called two-dimensional gel electrophoresis. All right? Two-dimensional gel electrophoresis. This one, I think you'll find is kind of magical. Okay? All right. But to understand it, we've got to understand something about proteins and charge. So I hope that you recognize that proteins can have a variety of charges based on what kinds of R groups they've got, what pH we're at, etc., etc. Okay? We have a technique that is called isoelectric focusing. Isoelectric focusing. This is a technique that allows us to separate proteins on the basis of their charge. It allows us to separate on the basis of their charge. I'm not going to tell you how it works because it may confuse you. All that really matters for us to understand is it's separating them on the basis of their charge. Okay? I can take a mixture of proteins. I might take a bunch of cells and I take all of the proteins from those cells. Not just one, but I take all the proteins from those cells and I mix them all up and I mush them all up and I've got them all in one thing I'm getting ready to load. Okay? And I take that mixture of proteins that has all the proteins of a cell and I put it into this apparatus called an isoelectric focusing apparatus. And it basically is just a tube that has sort of a gel-like material in it. Okay? At one end of the tube, the most negative proteins will be. At the other end of the gel, the most positive proteins will be. And in the middle, we'll have gradations, 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 till in the very middle we'll have charges of 0. Are you with me? So we've got this tube, and it's got all the proteins of a cell, and these proteins, positive, negative, or most negative out, most positive out, and then gradations all the way in. So we've got kind of a spectrum of charges of proteins. So a particular protein is going to be found at one or another place based on what charge it has. Everybody with me? Okay, let's say we take that tube that has this little gelatinous stuff, and we now put it at the very top. We cut it out, and we take this whole tube, and we put it at the top of a polyacrylamide gel. And we add some SDS. We didn't add SDS to separate. We separated on the basis of their charge without the SDS. Now we're adding the SDS to it, so all those proteins that had individual charges are now going to all get coated like before, and they're going to be sitting in this tube on top of a polyacrylamide gel. Can you envision this? Now I run gel electrophoresis, and I start separating on the basis of size. So first I've separated on the basis of charge, and then I coat them with SDS, and I separate them on the basis of size. You with me? The two dimensions, our first dimension was separated by charge. The second dimension was going through the gel, running on size. Okay? Now, what I get when I do that is something really amazing. Okay? This is a simplified diagram of what's actually there. Okay? The simplified diagram shows, first of all, that we separated this guy from left to right, most positive over here, most negative over here. Zeros right in the middle. And then we ran down this way, separating on the basis of size. The proteins that are the biggest and had the most most positive are going to be up here. Those that were the most negative and the smallest are going to be over here, and various other things inside of here. Well, why do we care about this? This is amazing, because what I can do is I can take all of the proteins of a cell, and I can separate them on one gel, and every spot that I see on that, and this is a simplification because most of them will have thousands of spots, every spot I see is a unique protein. The intensity of the spot will vary with how much of the protein. A very intense spot means more protein. Less intense spot means less protein. I've just described to you one of the coolest tools that you can have for doing research on proteins. Okay? How many have ever heard of the term proteomics? Okay, a couple of you. Proteomics is the ability to analyze all the proteins of cells simultaneously. Why is that powerful? Well, I'll tell you. Let's imagine I've just run this gel, and I look at it and say, oh, that's really cool. I see all these spots, and what the heck am I looking at? Right? What the heck am I looking at? Well, let's imagine instead of running one of these gels, I ran two. Let's imagine that the one that I ran over here were all the proteins that I isolated from, let's say, some cells of my skin. I took skin cells, I busted them open, and I analyzed those skin cells, and I have all the proteins distributed across here. And on the other gel over here, instead of using my skin cells, I took cells from a skin tumor that I have. And I separated them in the same way. Oh, I got something cool. Because now I can compare these two gels, and I can say, look, this spot over here is present in the normal cells. It's not present in the tumor cells. And look, this protein over here is in the tumor cells, and it's not in the regular cells.
Is that a way also to draw the phylogeneticity between the different organisms?
It's not a way of doing phylogenetic. No, it's a way of comparing within a given batch of cells what proteins are present in one versus the other. Phylogenetic analysis is different, okay? This is really amazing, because now I can say, here are all the proteins, and by the way, I can easily determine which protein corresponds to which spot. I can say, here are all the proteins that are found in a tumor cell that are not found in a normal cell. Here are all the proteins that are found in a normal cell that are not found in a tumor cell. Here are the proteins in the tumor cell I might want to find a strategy for attacking, or I might want to find a strategy for doing something about. I have just found a way to tackle cancer. Yeah? Yeah. So his question is, can you compare, how much can you compare densities? That is, the darkness of a spot, how much resolution is there? Within an order of magnitude, 1 in 10, you can easily tell. But less distinct gradations than that, it would be hard to do much the quantitative analysis. Okay? But the point is that you would be able to make distinctions between those two types of cells. It doesn't have to be cancer. You could say, well, hey, I'm interested in, I work for a pharmaceutical company, and I just created this new drug, and I think this new drug is the cat's meow. I wonder what the effect it is on the regular cells of the body. I could take and I could run a 2D gel and compare untreated cells, and I could take and run another 2D gel, and I could compare treated cells. And I could know very quickly, is there a difference in the expression of proteins between the treated and the untreated? And moreover, what are those proteins? Are they something I should be concerned about? Are these the same proteins that are popping up in a cancer cell? 2D gel electrophoresis is a phenomenal technique that allows us to make these kinds of comparisons. It's revolutionized how we analyze proteins. Absolutely revolutionized how we do that. Questions about that? Everybody's ready to get the day over, aren't you? Yeah. So this technique is probably less than 20 years old. It's getting improving with time, and it's getting much more reproducible with time, so that we actually can make these comparisons quite readily. Yes, question? I'm not sure I said the question.
You use the isoelectric focusing to find protein or a way to specifically use that protein to another substance in the cell or body without using that protein.
Shh. I can't hear what you're saying.
Would you be able to use this to find a way to attach protein to, say, a carbohydrate?
No. So this wouldn't help you with being able to attach a protein to something else. No, that would actually involve some chemistry and would be a little bit different. Yes? Very good question. So if I see a difference and I say, wow, look at this big spot over here in the tumor and it's not over here, I'd sure like to know what that is and what's there. You bet you can. Because people have done that and because these are so well characterized, we actually can look at a spot and know its XY coordinates and basically tell what it is. But yes, we could do that. We might say, this is kind of interesting. I wonder, for example, maybe getting back to your question over here, I wonder, this protein's here, but I wonder if it's been modified in some way. Does it, for example, have a phosphate put onto it? And so extracting that from the gel and doing that kind of analysis, you betcha you could do. And people do this a lot. Very cool stuff. Okay, so this business of purifying proteins really tells us an awful lot of very cool information. Okay, I'm going to skip over HPLC, and I'm only going to talk very briefly about this down here. Proteases. We're going to talk about proteases later when we talk about how enzymes work. But I want to introduce the topic of proteases to you. A protease is an enzyme that breaks down a protein. And it breaks it down by cleaving peptide bonds. A protease will cleave peptide bonds. All right? Here's a commonly used protease called trypsin. Many of the proteases, by the way, come from your digestive system. Your digestive system has proteases to break down the proteins that you have in your diet so you can take those amino acids and do something with them. Okay? Trypsin is a protease that cuts not just every peptide bond, but only specific peptide bonds. It cuts specifically peptide bonds adjacent to where there is an arginine or a lysine. Anybody recognize the similarity in why those two might both be of interest? Why it would cut next to both of those? What chemical property they have that might be similar? They're both positively charged R groups when they gain a proton, right? They both have a mean R group, so we call them basic. Yes? Okay? So chemically, they behave similarly, and the enzyme recognizes that, and it cuts adjacent to them. That's the only place it cuts them. It's very specific at where it cuts. Most proteases are fairly specific about where they will cut. Well, that means if I take a protein that's very large, and I want to make smaller pieces, that smaller pieces make it easier for me to analyze, I could actually chop it up with something that cuts in specific places, and I will get some specific fragments from that. So proteases allow me to take bigger, more complex proteins and break them down. That's what it does. Make sense? Okay, so trypsin cuts adjacent to lysines and arginines. We use other reagents in the laboratory that cut in other places. Chymotrypsin, I'm not going to make you know all these, but it cuts next to several places. It cuts adjacent to tyrosines and tryptophans. It likes the large phenolic groups that have the benzene rings. You don't need to know that. But suffice it to say, it also has some very specific amino acids it cuts next to. It will cut next to 5 or 6 different ones. But again, we get a specific set of fragments that are there. Another reagent we use to break down proteins is called cyanogen bromide, and it's not an enzyme. It's actually a chemical. But we use it because it's a chemical that breaks proteins wherever there's a methionine. Wherever there's a methionine, it breaks it at that place. So we can see here's something that's got a methionine. We add cyanogen bromide. You don't need to worry about the mechanism. But the bottom line is that we get 2 pieces out where we had 1 piece before. And that break occurs right where the methionine was. Okay, the methionine actually gets altered in the process. Okay. Now, that's what I'm going to say about protein purification. I'd like to turn next to talking about enzymes, but before I do that, I'll take some questions because I'm kind of moving kind of fast here. If you don't slow me down, I'll just keep going. Yeah. Yeah, the first protease, the trypsin, do I want you to know where it cuts? Yeah, I think you should know where trypsin cuts. And it's very simple. It cuts the 2 basics. It cuts next to the lysine and arginines. Yeah. Good question. Which side of them? Okay. I wasn't going to expect you to know that, but I'll tell you because you've asked the question, and no, you still won't need to know. It cuts on the carboxyl side. Okay. There are sides. You're exactly right. It could cut on the amino side, or it could cut on the carboxyl. It cuts on the carboxyl side. Yeah. Yeah, that's a good question. Is there anything different about methionine? Well, it turns out there's something different about every amino acid because they all have different R groups. So, I'm sorry? So methionine is the first one used in DNA synthesis. That's unrelated to what's happening with cyanogen bromide. Okay? So cyanogen bromide is actually picking on the unique chemistry of the side chain of methionine that the others don't have. So there's not anything chemically different about methionine that makes it be the first excuse me, amino acid that's used in protein synthesis. But let me save talking about that until we get to protein synthesis, okay? Other questions? Good questions. Friday afternoon, people are asking questions. People are awake. You can tell the exam's coming, right? That's good. Okay, let's talk about enzymes. We've been talking around enzymes. Now let's talk about enzymes. Enzymes are, as I talk about, critical for life, okay? We have to have enzymes in order for us to function. Now, I'm going to tell you a couple things about enzymes that are going to blow your mind. They blow my mind every time I say it, okay? Enzymes speed up reactions. We talk about catalysts. You learned that in basic chemistry. Catalysts are things that speed up reactions, okay? That's one of those things you had to memorize in general chemistry. Enzymes speed up reactions. And they came and they asked you on an exam, and they said, what do catalysts do? And you wrote down very dutifully, you said, enzymes or catalysts speed up reactions. Well, enzymes are catalysts, and they speed up reactions. Duh, right? Well, I'm going to give you a really amazing thing about enzymes. Chemists talk about catalysts because chemical catalysts are not biological. That is, you might add platinum to a reaction, and platinum might make a reaction go 100 times or 1,000 times faster. And 100 times or 1,000 times faster is pretty good because if you want that reaction to go, you want to make it happen. Living organisms have incredible needs for speed. Incredible needs for speed. We'll talk about some things later that will blow your mind. One of them is, we go replicating our DNA. We have little motors that replicate our DNA, and they operate at 6,000 RPM. 6,000 RPM. Something's got to power that. Something's got to happen very rapidly at the nanoscopic level. Okay? Things in cells have to happen very, very fast. Enzymes are therefore absolutely necessary because enzymes are making the things that allow 6,000 RPM reactions to happen. Okay? An enzyme, I said a good chemical catalyst might speed up a reaction 100 or 1,000 times. An enzyme is a biological catalyst. It is a protein. And there are enzymes that will speed up reactions, I am not making this up, over 100 quadrillion times. You might wonder, how many is a quadrillion? A quadrillion is 1,000 trillion. A trillion is 1,000 billion. A billion is 1,000 million. There are enzymes that are speeding up reactions 100,000 trillion times faster than they would occur without the enzyme. Now, you want to know why people want to study enzymes and make things happen rapidly, and why industries and so forth are interested in enzymes? The reason is because enzymes do the seeming impossible. They do magic. They do magic. Now, I don't believe in magic. I really don't. But I think enzymes come about as close to magic as it gets. I gave a lecture this morning entitled My First College Lecture to a group of students that were on campus visiting as a way of visiting OSU. And I told them a story that I'll tell you, and it's true. We have an enzyme in our blood called carbonic anhydrase. Okay? Carbonic anhydrase is an enzyme that will take and it converts carbon dioxide and water into carbonic acid. Okay? And when we look at enzymes, we compare the rates with which they work. Okay? We say, how fast does an enzyme work? All right? A good enzyme might catalyze maybe a few hundred reactions per second. If you think about catalyzing a few hundred reactions per second, that's a pretty amazing thing. Because the things that have to get catalyzed have to diffuse through the water. They have to get into the enzyme. The enzyme has to do their thing. It has to let go of them. They have to get away, and another one has to come in. And thinking of a few hundred of those per second is pretty darn amazing. Carbonic anhydrase blows your mind. Carbonic anhydrase will catalyze 1 million reactions per second. Every enzyme. Okay? You put one enzyme in there, one single enzyme molecule, and it'll catalyze 1 million reactions per second. How in the world does that happen? This enzyme works so fast that the only thing that limits it is the rate with which things diffuse through water. That's the slowest component. Enzymes are magical. All right. Well, I hope I've convinced you enzymes are magical. Now I want to say a little bit about how enzymes work. Okay? We've got to understand a little bit about how enzymes work, and we're going to spend just a little bit of time talking about energy. How many people like talking about free energy during freshman chemistry? Everybody, right? What? Well, let's talk a little bit about it here anyway. Okay? I'm going to keep it simple for right now. Okay? Got to keep it simple. All right. I'm going to show you a plot here. All right? The plot shows you free energy on the y-axis, and we show the progress of a reaction on the x-axis. Meaning the further we go to the right, the further the reaction is going. It shows us, from an energy perspective, what happens in a chemical reaction. In a chemical reaction, we have a so-called activation energy. That activation energy is an energy that must be overcome in order for the reaction to get started. You can see it. Here's the energy of the starting material, known as the reactants. The reactants for an enzyme we call substrate. Reactants for an enzyme are known as substrate. The energy of the substrate is right here. 2 substrates. Here's the energy of activation. This is how much energy we have to get up to. And then the reaction happens, and it falls down over here, and here's the energy of the products. Notice that the energy of the products is lower than the energy of the reactants. Energy is released in the process. There is a net release of free energy. Okay? Here's the same reaction with an enzyme. Notice that we have a lot lower energy of activation. But notice that the energy of the reactants and the energy of the products did not change. Enzymes do not change the overall energy of a reaction. They only affect the activation energy. The activation energy turns out to be the biggest limitation in the rate of reaction of an enzyme. Of a reaction. The activation energy is the rate limiting thing for how fast a reaction goes. If we lower the activation energy, we can increase the rate of the reaction enormously. Yes?
Does that mean that we need this energy if we have an enzyme?
Does it mean what?
We need this energy when we have a catalyst?
No. She said, okay, so that's an important question. She said, does it mean we need less energy if we have enzymes? No, because we haven't changed the energy overall at all. We have the same energy at the end as we had in the uncatalyzed reaction. All right? Only that getting the reaction started, but in the end, we're still at the same place that we were, so we don't need less energy. We don't change anything about the overall energy picture, only the energy necessary to get the reaction started. There's no net change in energy. Let me give this to you in another term. Let's imagine, if you will, that as a class we decide as a project, we are going to take this giant ball bearing. And let's say the ball bearing is about the size of this room. So we're going to take the whole class, and we're going to push this ball bearing to the ocean. Well, we can say, all right, well, let's think about this. Let's try to do it the easiest possible way. So if we think about doing it the easiest possible way, somebody gets out, and they say, well, we are at about 200 feet above sea level, and sea level is, of course, at 0 feet. So if we push this ball to the west, it should go into the sea. Well, of course, it doesn't. And the reason it doesn't is that there are energy barriers along the way. So we say, well, we want to make sure it goes to the sea, and we're assuming we don't have all these trees in the way, which if we keep clear cutting, we won't, right, I guess, right? So we assume we don't have anything that gets in the way. We want to find the way that we ensure it gets over to the sea. So what do? Well, as a class, we say, well, it's going to be our project. We're not going to lecture today. We're not going to sing today. We are going to take this ball bearing, and we're going to push it to the tallest mountain near here, because we know if we get it to the tallest mountain, it'll have enough energy. It'll go down, it'll go up, it'll go down, it'll go down, it'll finally make it down to the ocean. Right? And you'll ensure that it gets there. But then there's going to be somebody here who's going to say, man alive, we're going to spend all day long pushing that thing up the tallest mountain. Is that the stupidest thing we could possibly do, or what? All we have to do is get it over the lowest pass. We push it over the lowest pass, it makes it over to the ocean. Right? That's what an enzyme's doing. It's finding the pass. It's not going over the highest mountain to get over there. It's going over the lowest pass. We save energy. We have more time. We can come back and learn about biochemistry. That's good. You guys are dead today. It's like Monday. Right? So that's what an enzyme is doing, is it's finding the pass. It's finding the shortcut that's going to get us over there and get the ball bearing over to the ocean. Okay. Here's a comparison of catalysts that I was talking about before. Here's a platinum catalyst. Okay. We see the relative rate that a platinum catalyst has. In this case, it has an overall rate of about 27,000. This guy has a rate here of 650 million. This is just one catalyst, okay? The comparison, of one enzyme. One enzyme compared to one catalyst. Biological enzymes, biological catalysts are way more efficient than chemical catalysts are. Way more efficient. So again, it reiterates what I'm saying with respect to why enzymes are important. Enzymes allow us to produce energy when we need it rapidly. They allow us to mobilize and actually to respond to environmental change very, very rapidly. So we can handle those things because we have enzymes. If we don't have enzymes, we're in trouble. Okay? Temperature affects enzymes. And it affects enzymes different than it affects catalysts. Look at this plot. This is showing the maximum activity as a function of temperature, and we see that it goes up very nicely, and then it comes back down. Why doesn't it keep going up? What's that? The protein is denaturing. Remember, enzymes are proteins. What happens when we heat a protein up? We start denaturing it. When we denature it, da-da-da. Famous experiment, okay? Scientist studying fruit flies. Scientist has the idea that fruit flies behave like an ideal gas because they move randomly. You guys have heard this before. I see some smiles, all right? They behave randomly. They move randomly just like the molecules in an ideal gas does. So they set up this camera, and they point it into this chamber where they've got fruit flies. And they've got hundreds of these fruit flies in there, and they ask the question, do they really behave like an ideal gas? So they monitor the movement of these fruit flies, and they raise the temperature 10 degrees, and they see that, in fact, as they raise the temperature, the fruit flies fly faster. And they raise it 10 more degrees, and they fly even faster. And they raise it 10 more degrees, and they fly even faster. And they raise it 10 more degrees, and they stop flying. And they fly. They do that because fruit flies are just like enzymes. Yes?
So this is what happens kind of like in our body when we have a fever, right? It's not good.
So fever in our body can, as we talked about before, affect some enzymes. That's correct. So fruit flies only behave as an ideal gas up to a certain point. After that, you've got problems. Okay. Let's see. Let's talk about how enzymes work. All right? So I've told you from an energy perspective how enzymes work. Now we're going to talk about how enzymes work from a very different kind of a perspective. And this is going to introduce something we've been talking about with respect to proteins that I hope is now going to start making some sense to you. All right? If you take a basic biology class, and you've taken basic biology classes, they probably told you and showed you something like you see on the screen. That is that how many of you have been told that enzymes and substrates are like a lock and key? Everybody, because that's what they tell you in biology class. And you know what? It's completely wrong. You should go and demand your money back for them telling you wrong stuff. It's wrong. Okay? Now, there are some things about enzymes and substrates where there is an analogy to a lock and a key. But that is not the way in which they work. So what does that mean? Okay. Let's go back to the beginning. The beginning was, this was the way that people thought enzymes worked. They thought that enzymes bound their substrate in a very specific way, just like a key fits into a lock. Certain keys won't fit into locks because they've got the wrong dimensions, et cetera, but you get the right key and the right lock, you turn it, and you unlock the lock, and you're set. That was the thinking about how this worked. But people, when they started thinking about it, said, well, this tells us an awful lot about how an enzyme recognizes a substrate, but it doesn't tell us anything about how the enzyme actually works. How does the enzyme work? Okay. How does the enzyme convert the substrate into something else? How does the enzyme have this magical effect that's greater than what chemical catalysts have? Okay. Well, this brought about another description called the induced fit model that's much more accurate for how enzymes work. And I know this looks similar, but I'm going to tell you that it's not. And I'm going to explain to you what the differences are. Notice on the left, we have an enzyme that has 1, 2, 3, and we have point, point, point. And those points match up just like lock and key. They fit in, etc. It tells us how this enzyme fits in there. I want you to look over here at this guy. We have point, point, point. But over here we have 1, 2, 3. And they're not point, point, point. They are, in fact, rounded. The enzyme, it turns out, does not start out exactly like the substrate. It doesn't fit the enzyme exactly like a substrate before the substrate comes in. Now, well, what happens? Well, as the substrate is coming in, the substrate has some chemical characteristics of its own. It might have charges. It might have some chemical groups that are sticking out. It might have a variety of things, and as they're coming in, they are entering the environment of this enzyme, and they're going to start doing what as they get closer to the substrate? What did 2,3-BPG do? It changed the shape of the enzyme. I'm sorry, the hemoglobin. Hemoglobin is not an enzyme. It changed the shape of the hemoglobin. This substrate, as it's coming in, is going to start causing the enzyme to start changing shape. And guess what? By the time it gets there, it will have the shape of the substrate. Well, so what? Why is that any different? Well, there's a big difference. The big difference is that the enzyme in its natural state wasn't sitting there with points. It was sitting there rounded. Which state do you suppose the enzyme wants to be in? Rounded. That means that when it's sitting right here, this enzyme is under strain. There's strain. And when you put something under strain, what does it do? It tries to resist that strain. This complex right here, this enzyme really wants to do something with this so it can go back to where it was. That strain causes this substrate to feel the strain as well. And because the substrate feels the strain, it now gets chemically altered. That's what's happening during catalysis. Strain is causing chemical stress that changes chemical reactions. This might happen in a variety of ways. I've shown it with one molecule. We might have two molecules. Okay? Many enzymes work on two molecules. I'm going to show you a cool example from one with my hands. Okay? There's an enzyme called hexokinase that we'll study when we talk about glycolysis. And hexokinase it binds to two different substrates. It binds to ATP and it binds to glucose. Okay? And during the reaction that's catalyzed by hexokinase, one of the phosphates from ATP is transferred over to glucose. Okay? You with me? That's the reaction that it catalyzes. Okay? Now, here's what happens when hexokinase works. It binds two things. All right? It's sitting here. It's wide open. All right? Along comes an ATP. It grabs it. Okay? This causes a slight change in the enzyme, not only up here where it grabbed to the ATP, but also in the overall enzyme. It started here, and now it's here. Next, hexokinase encounters a glucose, and it grabs it here, and then something really big happens. The enzyme goes through a shape change, and these guys come together like this. What did I just enable? I've just enabled the transfer of the phosphate to occur much more readily because they've been brought into close proximity. And then the transfer happens because of this strain. When the transfer happens, now I've got different molecules that I had before. And now that the enzyme no longer holds them, it no longer wants them, and it spits them out. Pretty cool. The reason that the enzyme is different than a chemical catalyst is because an enzyme is flexible. Enzymes are not rigid. Enzymes are flexible. You saw this with hemoglobin at a very limited extent. With enzymes, which are catalyzing reactions, we see it to an even bigger extent in some cases. And this flexibility enables this process to occur. Chemical catalysts like platinum don't have that option. They are not flexible. Enzymes are flexible. Yes?
What do you mean by affinity of that protein in an enzyme?
What does it mean to have a protein to have an affinity for a molecule? Well, we'll talk about that next time, but affinity basically means its desire to bind to it. So we talked about hemoglobin's affinity for oxygen. It was its desire to bind to oxygen and how tightly it holds on to it. Yes, sir? Does the strain affect the overall activity of the enzyme? That's a very complicated question. So I will say yes, but it's not something that we can discuss here. Okay? So the nature of the strain will affect the rate of the enzyme and the rate with which it binds and releases things. So it's kind of a complicated picture. So yes, the answer is the answer to your question, but it's more than we could talk about here. Okay? But very good question. Yes? Built on what? Built-in enzyme? Sure. You can go and you can design an enzyme from scratch if you'd like. Have they ever made a very good one? Not really. What people do, and more commonly, is they will modify existing enzymes. Okay? Enzymes are so good because they have evolved that way. Selection process has made enzymes more and more and more efficient. Okay? That's much more than we're going to do in saying, well, I think we should make this and then this and this and this and this. We might make something, but we're not going to have that evolutionary process that's produced the enzymes that we have. Yeah. And people are doing a lot of engineering of enzymes right now. A lot of engineering. Yes? Chaperones do not act as enzymes, no. Chaperones act primarily as a chamber to allow folding to occur. Okay? All right, everybody's getting restless. I thought we should maybe finish with a song. What do you guys think? Okay. This is a fun song. We're not going to cover any specific biochemistry with it, but I think you'll enjoy singing it. It's to the tune of She'll Be Coming Around the Mountain When She Comes. The anthem for BB 350. Everybody ready? Oh, the students taking BB 350, 350. Have an awful lot of things that we must know, 350. With acetic acid buffer, Kevin Ahern makes us suffer. The exams could not be tougher, 350, 350. There's amino acid side chains to recall. And the things it takes to make cholesterol. Anabolic, catabolic, Kevin Ahern's diabolic. I'm becoming alcoholic, 350, 350. There must be a way to jam into my head all the metabolic enzyme names I dread. Can you help me learn the places where the endonucleases cut the DNA in places, 350? I must find a way to make a better grade, or my GPA will truly get waylaid. I shall overcome frustration to achieve my aspiration on the last examination, 350. Here's the plan I made to help me to succeed. Fill the note card with the knowledge I will need. I put all of Ahern's quotes along with what each one denotes onto a massive stack of notes for 350. Though there's just one teensy problem I must fix. It requires some very skillful penman tricks. Squeezing info I must store onto the card he gave before will mean a font the size of 0.14. Okay, that last is just repeating the first line. So that's actually referring to the fact that on my final exam, you actually get to bring in a note card that I give you. And you can put as much information as you want onto that one note card, and students write incredibly small stuff. So, all right, see you guys on Monday.